Your intestine is one of the fastest renewing tissues in your body. The cells lining it are constantly damaged by food, digestive chemicals, microbes, inflammation, and normal wear. Yet the barrier survives because stem cells hidden deep inside tiny structures called intestinal crypts continually rebuild it. Then aging changes the system. Those stem cells divide less effectively, their ability to regenerate tissue declines, and when the intestine is damaged, recovery becomes harder. A new 2026 Nature Aging study may have found one reason why. The problem may not begin inside the intestine, it may arrive through the blood. Researchers connected the circulation of old and young mice. After exposure to the older animal's blood environment, intestinal stem cells in the young mice began behaving more like old stem cells. Their regenerative ability declined. Then scientists traced part of that effect to a familiar inflammatory molecule called tumor necrosis factor, or TNF. TNF activated a receptor called TNFR1 inside intestinal epithelial cells. That signal disrupted mitochondrial metabolism, reduced the cell's ability to burn fatty acids for energy, and weakened stem cell regeneration. Even more surprisingly, several interventions partially restored function. The study suggests that at least one part of intestinal aging may not be permanently written into the stem cells themselves. It may be driven by inflammatory signals circulating through the body. To understand why that matters, consider what the intestinal lining has to do every day. The inside of your gut is not technically inside your body in the same way your liver or brain is. It is a massive boundary between you and the outside world. Everything you swallow passes along one side of that boundary. Nutrients must cross it. Water must cross it. But bacteria, toxins, and harmful molecules must mostly stay out. The epithelial cells forming this barrier are replaced extraordinarily quickly. Actively cycling L-gear5 positive intestinal stem cells sit near the base of intestinal crypts and continually generate the cells required to replenish the lining. Aging reduces their proliferative and regenerative potential. Think of the crypt as a tiny construction site. The stem cells are the workers producing replacement material. When you are young, the construction crew responds quickly. Cells divide, specialize, and move upward to replace damaged tissue. As the crew ages, rebuilding slows. Scientists have known about this decline for years. What they did not fully understand was how much of it came from damage inside the stem cells and how much came from the environment surrounding them. The new experiment attacked that question directly. Researchers used a technique called heterochronic parabiosis. Two mice of different ages are surgically joined so they share part of their circulation. Their organs remain separate, their stem cells remain genetically their own. But molecules circulating in the blood can move between them. If a young tissue begins behaving old after being exposed to an old circulation, that suggests aging signals exist outside the tissue itself. That is exactly what happened. The intestinal stem cell aging phenotype could be transferred from old mice to young mice. Something in the older circulation was telling young intestinal cells to behave differently. This is the first major retention point in the story. The young stem cells had not lived for years. They had not accumulated decades of mutations. Their chromosomes had not suddenly become old, but their behavior changed because the environment changed. The researchers then looked for the signal. Inflammation became the prime suspect. Aging is associated with chronic, low-grade inflammatory signaling, sometimes called inflammaging. One inflammatory molecule in particular stood out: TNF. TNF is part of the immune system's normal emergency response. During infection or injury, it helps coordinate inflammation. That can be life-saving. The problem is persistent activation. When TNF remains elevated or repeatedly stimulates tissues, the same pathway that once helped defend the body can begin interfering with normal repair. The researchers treated aged animals with anti-inflammatory approaches, including antibodies targeting TNF. Intestinal stem cell function improved, but TNF alone was not the entire mechanism. Cells need somewhere to receive the message. That receiver was TNFR1. The team used young mice whose intestinal epithelial cells lacked TNFR1. Then they exposed those mice to the old circulatory environment. Normally, old blood weakened young intestinal stem cell function. Without TNFR1, much of that damaging effect was blocked. The signal could be present in the circulation, but the cell could no longer hear it properly. Then researchers asked what TNFR1 was doing inside the cell. The answer led to metabolism. Stem cells do not simply need calories, they need specific metabolic programs. A growing cell must decide which fuels to burn, how much energy to generate, and whether to use nutrients for maintenance or construction. In intestinal stem cells, fatty acid oxidation appears to be one important part of maintaining regenerative capacity. Previous research has shown that activating fatty acid oxidation can enhance intestinal stem cell function during homeostasis and aging. Fatty acid oxidation occurs largely through mitochondria. Fat molecules are broken down and converted into forms that can enter energy-producing pathways. You can imagine the mitochondria as power plants, but in a stem cell they are more than generators. Metabolism helps determine what the cell becomes and how strongly it can respond when tissue needs repair. In the new study, aged intestinal stem and progenitor cells showed reduced fatty acid metabolic programs. When young intestinal organoids were exposed to TNF, their ability to form healthy crypt-like buds declined. Mitochondrial pathways were disrupted, fatty acid oxidation fell, cell proliferation and regenerative capacity weakened. This produces a surprisingly simple chain: old systemic environment, more inflammatory TNF signaling, TNFR1 activation, weaker mitochondrial fatty acid oxidation, less stem cell proliferation, less intestinal regeneration. Inflammation was not merely sitting beside aging. In these experiments, it helped drive a specific metabolic failure inside the cells responsible for repairing the gut. But then came the more important question: could the process be reversed? The researchers attacked the pathway from different directions. First, they reduced inflammation. TNF targeting treatment improved stem cell function. They also used salicylate-based anti-inflammatory treatment. Fatty acid oxidation in aged intestinal crypts improved. Then they targeted the mitochondria more directly. Mitochondria are not static objects. They constantly split and fuse. Fusion allows mitochondrial networks to combine resources, exchange components, and adapt to changing energy demands. When researchers promoted mitochondrial fusion in aged intestinal stem cells, their function improved. That is an important clue. Even after aging had already affected the tissue, restoring part of its metabolic machinery could improve regeneration. The old stem cell was not necessarily broken beyond repair. Its operating conditions had changed, but this is also where the study can easily be misunderstood. The experiments do not mean aspirin is an anti-aging drug for the human intestine. They do not mean people should take TNF-blocking medications to keep their gut young. And they definitely do not prove that suppressing inflammation as much as possible is healthy. TNF is essential for immune defense. Drugs that block TNF are already used clinically for conditions such as inflammatory bowel disease and rheumatoid arthritis, but suppressing this pathway can also increase susceptibility to infections and produce other complications. A treatment appropriate for someone with severe inflammatory disease is very different from giving the same drug to a healthy person for longevity. The salicylate experiments were mechanistic evidence in mice. They were not a human anti-aging trial. There is another important limitation. Parabiosis is an extremely artificial experiment. Humans do not share circulatory systems with older people. The purpose is to isolate the effects of blood-borne signals. It shows that something in the old systemic environment can influence young tissue. It does not prove that TNF is the only circulating factor involved. Aging blood contains changes in hormones, metabolites, immune molecules, and many other proteins. Some may accelerate aging, others may protect against it. The intestine itself also changes with age. Its microbiome changes, local immune cells change, the stem cell niche changes. Paneth cells and surrounding stromal cells change. DNA damage, epigenetics, and cellular metabolism all contribute. TNFR1 is therefore one pathway inside a larger aging network. Still, the finding connects two ideas that aging researchers increasingly encounter across different organs. The first is systemic aging. An organ does not grow old in isolation. Blood, immune cells, hormones, and metabolites connect every tissue. The second is metabolic aging. A stem cell may still be alive and genetically intact, yet lose function because it can no longer generate or use energy in the correct way. In the intestine, those two ideas appear to meet. Systemic inflammation changes cellular metabolism. Cellular metabolism changes regenerative capacity. And regenerative decline may ultimately change tissue health. Why should someone care about intestinal regeneration if they do not have digestive disease? Because the gut barrier affects much more than digestion. A resilient intestine absorbs nutrients and helps prevent microbes and inflammatory products from crossing into tissues where they do not belong. When regeneration becomes impaired, recovery from injury can become more difficult, and disturbances in the intestinal barrier can interact with immune and metabolic disease. This does not mean intestinal stem cell aging causes every age-related illness, but the gut is one of the body's largest interfaces with the environment. Keeping that interface functional matters. The most interesting possibility from the new research is not a drug, it is reversibility. A young intestine exposed to an old environment became older in behavior, blocked the inflammatory receptor, and much of that effect was prevented. Reduce inflammation, and metabolism improved. Improved mitochondrial dynamics, and aged stem cell function improved. The aging phenotype was not completely fixed. That changes the question. Instead of asking why intestinal stem cells inevitably fail with time, researchers can now ask which signals are continuously telling them to fail. And if those signals can be identified precisely, perhaps some decline can eventually be interrupted before tissue function is lost. For humans today, there is no proven treatment that resets aged intestinal stem cells. The safest practical advice remains far less dramatic. Maintain overall metabolic health. Avoid smoking. Stay physically active. Treat chronic inflammatory diseases appropriately. Eat a nutritionally adequate diet that supports gut and metabolic health. And do not take anti-inflammatory drugs chronically without a medical reason simply because an animal study linked inflammation with aging. The science is not there yet, but the biological lesson is powerful. Your intestinal stem cells may not age only because they have existed for a long time. They may age partly because the rest of the body is sending them older instructions. In this study, one of those instructions was TNF. Its message traveled through TNFR1, reached the mitochondria, reduced fat burning and weakened regeneration, blocked the message, and the cells behave differently. Sometimes aging may not be a clock inside the cell. It may be a conversation happening throughout the body, and changing the conversation could eventually change how the tissue grows old.